if you would, tell me, if there was no control mechanism for cell division, what would happen? That's something, if you looked at the PowerPoints ahead of time, you would know the answer to this question. If there was no control system, what would happen? The answer is one word. Huh. We are talking about mitosis. You remember, you saw the video, the little boy was playing soccer, cut her leg. You know, now mitosis is happening. We are talking about mitosis. Bring you to the ballpark. You have a little boy or little girl at home, they are growing taller and taller and taller. Is that right? What processes are occurring to them? Well, mitosis. My God. You have a plant at home, right? Farmers planted some seeds. Now those plants are growing taller. And what processes are occurring in those plants? The mitosis. You guys are still sleeping. Mitosis. Is that right? Now, the question is, if mitosis keep occurring and occurring and occurring, there is no stop, there is no control system, what will happen? Cancer. You know about cancer, right? Did you wake up now? Or should I go get some coffee? Cancer. If the cells keep growing and growing and growing and growing, Houston, you watched Apollo 13 over the break? Houston, we got cancer. No, right? No, you watch Apollo 13? No, anyhow. So, there are 3 checkpoints based on your textbook. We are going with your textbook, what your Campbell said, recent Campbell said. There are 3 checkpoints, G1, sometime during you have G1, sometimes during G1 there is one checkpoint right here. And then when it comes during the beginning of the mitosis, right here is the beginning of mitosis, there is a checkpoint here. And there is a checkpoint also at M stage of mitosis. Okay, so M checkpoints, G2 at the end of G2, beginning of M, G2 checkpoints, and M checkpoints, and G1 checkpoints. Does everybody know what I'm talking about? Okay, so let's elaborate on those a little bit. Okay, there is a lot of research, as you know, people are researching cancer, your tax money, some of it is going for cancer. You donate, optionally, not your tax money, you donate some money to cancer research. This is it. That's what they're working on. And we are just putting 1 or 2 slides of PowerPoints into it. And people are spending their time, their life, working on cancer to figure it out, to how to stop it, at least. Okay, so 2 types of regulatory proteins are involved in cell cycle control system. Cell cycle control system. There are 2 proteins involved, which your textbook mentioned. One of them is cyclin. The term cyclin. Right, cyclin. Okay, cyclins and cyclin-dependent kinase. That word kinase, you've heard of. ASE is an enzyme. So the abbreviation for the CDK. But where else have you heard of this kinase word before? Do you remember what chapter, what we talked about in the past? No? You remember the protein kinase? We talked about them in the past, and the tyrosine kinase. You remember those cell membrane? That was a chapter on cell membrane. The term kinase, you've heard about it during the cell membrane chapter 11. Okay, so these 2 protein molecules, both of them protein molecules, are involved with control system, this control system, somewhere along the line. Let's see where they are involved. The activity of CDK, cyclin-dependent kinase, CDK rises and falls with changes in concentration of the cyclin partner. So these 2, based on that sentence, if you see a multiple choice question or essay question, these 2 are working together. Am I making some sense? So the fall and rise of this one, of cyclin, will turn on and off this one, CDK. Okay? And then MPF is a maturation promoting factor, is another molecule that is just by itself. They're not these two are related to each other. These two protein molecules are related to each other. The MPFK is a little bit away from these guys. It's a cyclin CDK complex that triggers a cell's passage past the G2 checkpoints into M. So MPF is maturation promoting factors. It goes from what passage past G2 checkpoints to G2 to M. So right here it would be MPF. Right in this area, that protein molecule is the maturation protein factor. It's a protein molecule. It's involved in that one. Okay, so this one is showing you by diagrams and charts and graphs how the cyclin and MPF are combined together. So that would be checkpoints, and CDK can separate from it. Cyclin is degraded. And then degraded cyclin is right here. So that shows you a little bit of how this diagram, this chart is going together with these two protein molecules. Again, this explains pretty much of what you read here. But look at it at home and trying to figure out how these two go together a little bit. And then right here again, he's beating the dead horse. G1, G0, when the cell is at rest, is called G0. I have not mentioned that in your PowerPoints, but make sure you know what G0 means. G0 means when the cell is not dividing anymore. A lot of times cells spend their time in G0. It means no division is occurring. But G1, G2, it means division. M, it means the cell is dividing. So G0, the cell is at rest, if you would. That's the best definition I have for G0. G0, it means the cell is at rest. So these cyclins and MPF are working to block the cell from going into G1. Okay, so without go-ahead signal, the cell enters G0, G1 checkpoints. With go-ahead signal, cell continues cell cycle, so that goes down and it will continue on and on and on. Okay, so the next few pictures again, they're trying to explain as what happens as the checkpoints of the cells. Okay, what is an example of internal signal? An example of internal signal is that the cells will not begin anaphase until all chromosomes are properly attached to the spindles at the metaphase plate. Okay, that's another checkpoint. This mechanism assures that daughter cells have the correct number of chromosomes. What is an example of external factors? External factors that influence cell divisions include specific growth factors. Growth factors are released by certain cells and stimulates other cells to divide. Platelets drive growth factors. PDGF is made by blood cell fragments called platelets. In density-dependent inhibition, crowded cells will stop dividing. I have to talk about that right here, and the next few PowerPoints will explain this a little bit. So there are two ways that the cells recognize how to divide, how to stop. One of the external factors. If I grab a plate right here, which that's going to show you, and I take a cell and put it in that plate, and there is a medium fluid in here, food for the cell, and the cells keep dividing. They keep dividing. Take one cell from your liver, for example, from any part of your body. And here are the nucleus. The cells keep dividing and divide and divide. And as soon as they touch a hard surface, the walls of the plate, and as soon as they touch each other, right here, then they stop growing. I'm going to make you some sense. The cells will not grow anymore. This is the normal case. Everything is normal. Now, what happens if you grab a cell, same thing, medium in here, food for the cell. You grab a cell and you place the cell in a plate, and they grow and grow and grow, right? They're supposed to stop now, but they will not. They keep growing on top of each other and keep growing. Of course, there is no space out here. If this was a membrane or some tissue, they would keep growing in there too. You all know what I'm talking about. Okay, but since this is a hard surface, they cannot grow anymore. They grow on top of each other. So then you know you're dealing with a cancer cell. Then you know you're dealing with cancer cell. Then this is cancer, or we call it abnormal. That's what cancer is. Cancer, in 2 words, it means abnormal growth. That's the best definition of cancer you're going to get anywhere. What is cancer? Cancer means abnormal growth. Okay, so that's what it says. A sample of human connective tissue is cut up in small pieces, and then of course enzyme digest the extracellular matrix, releasing the unsuspension of the fibroblasts. Fibroblast is a type of connective tissue. It's a cell. It's a type of cell. Cells are transferred to culture vessels. PDGF, external factor. PDGF is an external factor outside of the cell, added to the half vessels, and here they are with PGDF, without PDGF, platelets, developmental growth factor, and here is a with PDGF, the cells keep growing and growing and growing. Here they are, and then how is cancer being formed? Anchorage, density-dependent inhibition. That's what this is called. This process is called density-dependent inhibition. Cancer cells exhibit neither type of regulation of their division. Right here, as I said, that's what pretty much this diagram is showing you. Cancer cells and density-dependent inhibition and anchorage. Anchorage, it means the cells anchor to the bottom of the dish, and then they keep growing and growing and growing. As soon as they touch each other and they touch a hard surface, they stop growing. Okay, in case of cancer cells, they grow on top of each other, right here. The whole thing, the whole dish becomes full of cells. That's in case of cancer. But this is normal. This is a normal case. Okay, I hope. A normal cell is converted to cancer cells by a process called transformation. And what causes transformation is up for grab. It's a lot of debate. Sunlight, the DNA, changes of DNA. Cancer cells that are not eliminated by the immune system form tumors, masses of abnormal cells within otherwise normal tissues. If abnormal cells remains only at the original site, the lump is called a benign tumor. What happens, you guys, we all of us, without exception, have cancer cells in our bodies. Okay? What will happen, our immune system, our white blood cells, you probably have heard about it, our immune system go recognize them and eat them. Okay? But the problem arises when our cancer cells cannot recognize them anymore, or they evade our white blood cells. The cancer grows when our immune system cannot recognize them anymore, so they go wacko. Now, when they go wacko, when they start growing and growing and growing, it's called a benign tumor. A benign tumor, as you all have heard, you probably know, is something that is not dangerous. They go ahead with surgery, they remove it, no problem. You don't have to do much of anything. Okay? But you have lumps in your skin, I don't know, you've heard of, you go to the doctor's outpatient right here, they remove that lump. That's benign tumor. Okay, I'm saying the simplest of all. Okay, but however, the other one is malignant tumor, which you've heard of, have heard. The malignant tumors invade surrounding tissues as well. You remember this? I did draw it for you. Not only they get out of their place and they go to surrounding tissues. They go all over the bodies. They call it metastasize. Metastasize, it means the cancer cells travel and they go to all over the body. When they travel by our blood circulatory system, by our blood vessels, and they evade invade our entire body, then it's called metastasis, or metastasize. Okay, so invading surrounding tissue and can metastasize, exporting cancer cells to other parts of the body where they may form additional tumors. Okay, so localized tumors may be treated with high energy radiation, which damage the DNA in cancer cells. But of course, to treat metastasized cancer, chemotherapies that target the cell cycles may be used. That's what they are trying to do. With radiation and with chemotherapies, they're trying to stop that cycle, the cycle that we are talking about. Sometimes that radiation he's talking about here, the radiation damages the DNA of cancer cells. However, it damages the normal cells as well. The most receptive cells that are damaged when you use chemoradiation is the ovary and testes. Okay, the ovaries and testes are being affected a lot during this radiation therapy that they're using. Okay, here they are. We have a cancer in the mammary gland. It's growing and growing and growing. Of course, when these cancer cells get to the blood vessels, I talked about that, and lymphatic vessels, and they can go to the rest of the body. They can go metastasize. That's the term. That's metastasize. It means the cancer cell spreads to the rest of the body. Okay, telophase and cytokinesis, anaphase. These are the stages of mitosis. I don't know why he's going back here. And I think that's the end of the chapter. So the end of the chapter quickly talks about cancer, and make sure you know a few things about it. I'm done with that chapter. Let's go to meiosis. So cancer cells, if I may, cancer cells, if I may ask you, it's by what process? Occurs by what process? Or maybe I'm asking the wrong question. The cell cycle. You will see another cell cycle here too, but which one? Huh?
You got like mitosis?
Mitosis. Cancer occurs because of mitosis. Cancer, it means abnormal growth. Is that right? Abnormal growth. Right? So abnormal growth comes from what process? Mitosis. Am I making some sense? You guys are turning your brain a little bit to my way, how I think. What is this picture of? Anybody?
It's kind of flagella.
Flagella, yes. That's what?
It's a sperm, probably.
That's a sperm. And what do you think this is? Huh? Egg. Sperm and egg. That's where you and I came from. That's where Brian's baby came from. Is that right, Brian? Your daughter, how old is she?
3.5.
3.5? Lucky you. Okay. So that's where now today's lecture, I'm going to talk about where that sperm came from, where that egg came from. Do that make sense? A little bit, everybody? Let's go over that. Okay. A gene, inheritance of genes, gene, gametes. You all know what gene is. A unit of inheritance is called gene. A gamete is called sperm, egg, pollen. Those are all gamete. What you saw here, that's a gamete. That's a gamete. Is that right? This is a gamete. This is a gamete. So humans have 46 chromosomes in their somatic cells. So what am I saying? You have 46 chromosomes in the cells of your eyes. You have 46 chromosomes in the cells of your skin. You have 46 chromosomes in the cells of your bones, in the cells of your muscle cells. All of your body, all of your somatic cells have 46 chromosomes. Then, what do you think, how many chromosomes are in sperm? 23. How many sperms are in the egg? 23. You should know those numbers. You're taking biology class. Amir, I thought I'd take biology class not knowing any numbers. I'm sorry to break the news to you. Once in a while in biology, you have to memorize the numbers. The numbers of human, you should know it. 46. Okay, so 23 here, 23 here. 46 combined together. What process sperm and egg come from? It comes from a type of cell division called meiosis. The term meiosis and mitosis are very close to each other. You forgive them. You forgive our great-great-great-grandfathers when they tossed up those terms. You forgive them. Those two terms are very close to each other. Unfortunate. Reno mix them up sometimes. Right? Just like this morning he said meiosis. No, mitosis. Cancer comes from mitosis. Meiosis is where the sperm and egg comes from. Meiosis in Greek means meiosis. In Greek means reduction. Mitosis in Greek means what? Mitosis in Greek means what? I said that before. Huh?
I said production.
No. No. What is it? Mitosis in Greek? The what? Thread. Very good. Mitosis in Greek. When the very first people saw those threads, which you're going to see it on the microscope today, when they saw it on the microscope, they said, ah, those are threads. They had no clue they were chromosomes. They had no clue what they were.
What year did they find
So
those?
they looked at them on the microscope and they said they're thread. They called them, in Greek, called it mitosis. And then when they recognized about the number of chromosomes being reduced in sperm and egg, they called it meiosis. Reduction. Meiosis. It means reduction. Reduction of what? Chromosome numbers. Okay? So, a gene specific position along a chromosome is called, its locus or loci is a plural, and locus is singular. Locus, it means location of a gene on a chromosome. When they map genes, gene mapping, this summer, you all can take this class. It's called Human Genetics. Very simple, easy A, online. They talk about mapping a little bit. You will see about mapping, and I will talk about it here a little bit too, not too much. But anyhow, it's a very simple, easy class. Hydra budding. Let's watch and see what it says. This is hydra. This is an animal. This is an animal, and it's dividing by budding. This is budding. Right here. Here is a bud. This will separate from mother hydra. This baby hydra, is dividing by budding, and it will sit on the bottom of the lake. Okay. So, set of chromosomes in human, a karyotype, it means when they map, they put, you will see a karyotype here. When they take your white blood cells and take they take the chromosomes out and they put it on a plate and take a picture of it and say, these are your chromosomes. That's called karyotyping. And you will see a picture of it. I should have put a picture of it here. Homologous chromosomes, or homologous, it means chromosomes that are alike. You have two sets of chromosomes inside of the cell, moving around. Me and Maricela. Maricela, can you put your arm right here? I wish you, no, you two. You, put your arms up, please. Cross them. You too, Reno. Okay, so these two are homologous chromosomes. They are moving around inside of the cell, and during the cell division, and I will talk about it, they are together. They are homologous. Me and non-beat are together, and we move around inside of the cell. Am I making some sense? So we are homologous. We are chromosome number 10. We are chromosome number 11. Am I making some sense? That's what is called homologous chromosomes. They are inside of the cell, and you will see it. I'll show you a picture in a minute. Chromosomes in homologous pairs are the same length and shape same length and shape. Am I making some sense? Same length and shape. And carry genes controlling the same inherited characters. Okay? A karyotype, I'm repeating myself. Homologous chromosomes, I'm repeating chromosomes, pair are the same length. So I try to move them around, but anyhow. Here is a karyotyping. They take somebody's blood, right? They take somebody's blood and they karyotyped it. This is karyotyping. So this is chromosome number 1, Reno and Maricela. This is chromosome number 2, me and non-Vit. Am I making some sense? So how many pairs do we have? We human, how many pairs do we have? 23. We have 23 pairs, which is translating to 46 chromosomes. If you count them, there are going to be 46 chromosomes. But how many pairs? 23. Out of these 23 pairs, one pair is called sex chromosome. Am I making some sense? The other 22 pairs are called autosomes. Okay, do I have it in here somewhere? Anyhow, it's going to come up. Autosomes, right here. Sex chromosomes and autosomes. Okay, so let's go back here. So you have 23 pairs. Is that right? One pair is sex chromosome. In this case, you have X and Y. So they took this blood from a man or from a woman? X, Y, huh? From a man. They took this blood from a man. If this was XX, what was this blood from? A woman or a man? A woman. If this was XX, it was from a woman, but they took this blood and they karyotyped it. You all are familiar with the term karyotype now. They karyotyped it, and the karyotype is showing XY, so that's the blood of a man. Okay? There are some abnormal cases which you learn that some people have 2 XX and a Y. Or some women have 3 X's, XXX. Those are abnormal cases. Or there are a variety of them, XYY. Okay, so there are some variation, abnormalities, which when we get to chapter, I think 14, we talk about it a little bit. But anyhow, that's a normal case. And these are homologous pair, homologous pair, homologous pair. Chromosomes that are same length. This chromosome and that chromosome are not homologous. Am I making some sense, everybody? These two are same length. They code for the same inheritable material, so on and so forth. Right, Ariel? Okay, let's go back to terminology. Sex chromosomes, XY, XX, XY is sex chromosome, X and Y. Autosomal chromosomes, the other 22 pairs, which are not responsible for determining the sex of an organism, is called autosomes. Okay, diploid cells, the cells of your eye, the cells of your muscle, the cells of your bone, they're all diploid cells. It means they have all 46 chromosomes. Diploid cells are 2N, we call it 2N. It's like this one. This chromosomes were taken from a diploid cell. It has all 46 chromosomes. Am I making some sense? But if you go back, this has only 23 chromosomes. Do you remember that? I talked about it. This has 23 chromosomes. So this is a haploid cell. This is a haploid cell. Easy to remember, half. Haploid, haploid, half. Half of the number of the chromosomes. Right here.
So it has
Diploid.
pairs.
Haploid has 23 chromosomes. Haploid.
How many pairs does it have?
23 pairs? How many? 23 chromosomes? How can you make it a pair? It's 20 to 11 and a half pairs. That's right?
Yeah.
That doesn't exist. 11 and a half.
Oh,
Pair.
Alright.
Going to make sense? So there is no pair. You cannot say it if it's an odd number, you cannot make it as a pair. Right? Pairs come always in even number, you know. Okay. Meiosis, it means reduction of the number of chromosomes. Haploid, 1n, they call it 1n. It means half of the number of chromosomes. Fertilization, what is that? You tell me. We already talked about it. Fertilization means what? Oh, nobody. My God. You guys don't know what fertilization? You never heard of fertilization before. Huh? When sperm enters the egg, the process of entrance of sperm to the egg is called fertilization. In fishes in general, fertilization occurs in the sea. Fish, male fish release sperm, female fish release the egg, they go into the water and they fertilize each other. They're missing out. Oh, Stephen's smiling. Okay. Zygote, it means what? Zygote means what? Fertilized egg. When sperm, here you have sperm, here you have egg. When they come together, the process is called nucleus. The process is called fertilization, the process. But after the sperm enters the egg, this one cell is called zygote. You need it for zoology, you need it for the rest of your life. For bio 2, bio 3, you're taking subsequent biology classes, that term will come up. Zygote means fertilized egg. It means the egg, the sperm that entered the egg. Now you have how many chromosomes? In case of human, how many chromosomes are in zygote?
46.
Huh? 46 chromosome. 23. Last time I checked my math. 23 plus 23. Catherine is 46. Okay, here is this is 2n. They're showing you 2n. It means there are 6 chromosomes. 1, 2, 3, 4, 5, 6. These two are homologous to each other. These two are homologous to each other. Those two are homologous to each other. Right? You all know what I'm talking about. Okay? So centromeres, they are already duplicated. They are connected together in a centromeres, a pair of homologous chromosomes, one from each set. Two non-sister chromatids. Okay? They're non-sister. These two, that is a non-sister, that is a non-sister chromatid of homologous pairs. Sister chromatids. This is a very nice slide. It will put everything in perspective to you, for you guys. Put everything in order. This is one of the essay questions I ask you. This is the life cycle of human life cycle. I ask you, what is the life cycle of human? This is it. Let's go over it. You all know it. But let's go put these terminologies that you know, put it in here. Okay? Are you guys ready? Let's go over it. Let's go over with the ovary and testes. Testes, of course, male, female ovaries. Another name for ovaries and testes is called gonad. You should know these. When I was playing flag football, did I tell you that story? Gonad? No. When I was playing flag football in Mississippi State I was there for one semester. We played flag football. The name of our team was Nads. Nads, and I asked my teammates, what does Nads mean? I said, Amir, you will find out first day of the game. First day of the game I went and cheerleaders were on the side and we were playing ball, and then the cheerleaders were cheering, go Nads, go. Then it clicked. Why the name of our team was Nads. And I still have the t-shirt. Number 8, always number 8. I can bring it to you guys. And we won, we were champions. Gonas became the champion of the league. And I have that t-shirt too. You play hard and they give you a t-shirt. Peyton Manning gets millions of dollars and
You get a t-shirt.
I just got a T-shirt, but still, it's better than nothing. They should say, they could say, oh, good job, gonads. Go home now. But there you go, it's a T-shirt. Okay, so the ovary and testes are also referred, the generic name for them is called gonads. Generic name. Okay, the general name for ovary and testes is gonads. So, where in our body meiosis occur? Right? In the gonads. Am I making some sense? So, ovary makes what? By process of meiosis, what does ovary make? No? Anybody paying attention? Is anybody paying attention? Nobody. Oh, I talk to myself. Like those crazy people, they talk to yourself. Okay. Huh? They make what?
Zygote?
No. Ovary makes egg. What does testes make? Huh? Sperm. By what process? Meiosis. Okay, sperm and egg. Now, fertilization occurs, right? And when fertilization occurs, zygote is results. Zygote in female, of course, in uterus of the female, the zygote undergo what process? Mitosis for 9 months later than you were born. Am I making some sense? After you're born, still mitosis occurs. At what age meiosis occurs? Age 1, 2, multiple choice question. At what age meiosis occurs? Age 1, age 2, age 3, age 4, age 15, age 10, 11. Meiosis occurs when you go through puberty. In case of females, 9, 10, males, 12, 13, somewhere in the neighborhood. That's when meiosis starts occurring. Meiosis does not occur at age 1, 2, 3, 4, neither one, male or female. 5, 6, does not happen until age puberty, which is different people, different regions of the world. People in closer to the poles, North Pole and South Pole, usually in colder climate, usually occur later. People who live near equator of Earth, I'm talking about Earth, near the equator, puberty occurs a little bit earlier. Some people in Siberia, puberty occurs at age 18. I doubt it nowadays, old days. 18, at age 18 they become mature. Meiosis. Females in Saudi Arabia at age 9. In the United States too. 9, 10, but anyway. Here is, I gave you a few shots of different type of, this is pretty much similar to human. This is in plants, and these are in unicellular, single cell, protozoa. So different type of life cycle occurs in different organisms. I gave you a shot at. Gametes are the only haploid cells in animals. They are produced by meiosis and undergo no further cell division. Sperm does not divide and becomes another sperm. Egg does not divide, become another egg. That's the end of it. End of meiosis is end of that cell, unless they become fertilized. If not, those cells disappear. What happens to female's egg if they are not fertilized? What will happen? They disintegrate. Other cells come and eat them. They disappear. You know that. Same as human sperm. Human sperm, if we don't, if the male does not release them, get them out of their body, then they vanish. They disappear. Other cells of the body in the testes will eat them. Okay, that's what happens. what did I say? Gametes fuse to form diploid zygote that divides by mitosis to develop into a multicellular organism, which we already talked about. Here's again. Plants and some algae exhibit an alternation of generation. That is used, that's a term used for plants. I try not to use it in zoology for animals, but some animals show that a little bit too. This life cycle includes both a diploid and a haploid multicellular stages. The diploid organisms called the sporophyte makes diploid spores by meiosis. Here is the alternation of generation. Alternation of generation examples. You have a plant that is undergoing mitosis, and you have a plant that is going under meiosis as well. Two separate plants. Okay. When you take botany, you will learn about it. I'm not going to talk about it too much in here. Here is a protozoa, flagellated protozoa that undergo that process as well. Study it. I'm not going to go over it too much. Okay, so let's go back to what you have to worry about and you have to know. As you know, we are going to go over meiosis. This is an overall picture of meiosis. Okay. One red chromosome, one blue chromosome. During interphase, a lot of textbooks miss out this part of the meiosis. So you have one red and one blue, and let's take a 5 minutes break. Let's do that because you guys, I want you to come back. It is important information. I want you to come back and